All right, now we're back for part 6, which is my favorite topic. What are the examples of cross-reactivity that we can see in the immune system? And I've got 5 examples for you. I know there's more, and in fact, my students, you have the chance to go and find more. Each of these examples is the kind of thing that I want to hear about if you want to tell me, find another paper like this. And so there's these whole issues, and I mentioned the case where the problem with using T-cell receptors is that everyone has a different array of MHC molecules, and T-cell receptors respond to the MHC molecules in the context of peptide. In fact, that seems pretty unavoidable. If you look at the size of the T-cell receptor and the size of the peptide, yeah, it's centered on the peptide, but there's a lot of the alpha helices on either side that it's going to bind to. But then people were looking at, what if you did some engineering so that you focused the T-cell receptor onto just the peptide itself, so it would bind only the peptide and not the alpha helices on either side. This would be very narrowly focused. Could you do something that was peptide-centric? In that case, you could actually have it respond to the same peptide in multiple different MHCs, because it wouldn't actually be touching the MHC. It'd just be touching the peptide. There is a company called Myrio Therapeutics that is doing exactly this, and they have made an engineered T-cell receptor that will just bind peptides, at least in mice. This is why this is called cross-HLA targeting. It doesn't care about the MHC. It just cares about the peptides there. And the peptide is related to an oncoprotein. That means a cancer, a protein upregulated in cancer that ends up in the MHC class 1 immune cells on the surface of the cell. So here's the peptide itself. This is the peptide. You can see the sequence right there, QYNPIR-TTF. And it comes from a protein called FOX2B, which is upregulated in neuroblastoma. So a bunch of these peptides get on the surface of the cell. The problem is, people have different MHCs, but the same peptide ends up on multiple MHCs. So what they've done is they've made a receptor that binds just the red stuff and doesn't bind the gray stuff here. We want a TCR that will bind just the peptide, not the MHC. Now, for the reasons that we've talked about, the antibodies are easier to work with, and that's true even at a structural level if you're doing this kind of protein design and engineering. This group actually made a TCR by taking the bottom part of a T-cell receptor and attaching it to the top part, making the top part an antibody's variable region. So they actually, because the antibody's variable region is so much easier to work with, and also you can focus that, it's not trained on an MHC at all, so you can focus that on a peptide much more easily. So technically this is a chimeric antigen receptor, not really a TCR. It's got the feet of a TCR, but its top, its head, and its chest are made of an antibody fragment. And they developed the antibody fragment, stuck this into a TCR, a T-cell, and then it turns out that it activates the T-cell. People do this, by the way, with whole T-cells, by the way, for other situations. But this is the first case that I know of where they've actually done it for a peptide-focused rather than peptide MHC-focused situation. Here's their picture of their idea. They put their antibody on different things, and they did screening, kind of like what Wells and the Antibot did. It's just they had a nice little screening technique where they would screen for things that would bind peptide always and MHC never. They did it against a bunch of different MHCs with the same peptide, and they eventually gave it, and they found out that it causes tumor killing, no matter what the different MHC molecule, the different HLA gene that it has in it. You see over here it has 3 different MHC molecules, 3 different HLAs, but they're all binding the same red peptide, and they're all reacting with the same PC CAR. Remember, CAR is the receptor. So here is their thing where they've shown that they have a receptor that binds just peptide. If you look over here on the left, it's another one of these binding Biacore type techniques, where the higher the line is, the more binding you have. You see that green line that's really high? That means really tight binding, because it goes up fast and it stays high. That green line is high. And so they have an antibody fragment that will bind to this thing, and they switch it out to different MHCs, and it always binds as long as it has the same peptide. In fact, they even did some alanine mutations that you see here, and you see that sometimes you mutate each of the 8 residues, you can mutate them each to alanine, and 1, 2, 3 times it doesn't care, but 1, 2, 3, 4 times it does care. It looks like they couldn't mutate residue 2. It doesn't matter anyways because it probably wouldn't do anything. But you see all the ones where it really cares whether it's alanine or not, they color those red, and those show up right in the middle of the molecule. If you look at part H, all these red residues means if you mutate it to alanine, you blow up the binding, the nice binding that they see. So it really cares about having these particular residues right here in the middle of the target. So in other words, this means the Tcell receptor is binding right where they expect it to, and they can even see that all those residues that should be important are indeed important. The other check to do is if you put this same peptide into different MHC contexts, will it still work? And they have 3 different MHCs, the gray one, the green one, and the yellow one. And they put them into all those, and it works for all of those. The green line that you see on the bottom for the Tcell that is moved more to the right on the left 3 columns that you see that the green line is moved over, and that means that you have binding of the Tcell receptor to these 3 different situations. However, if you look at the 4th column, you don't see binding there. And that's their deliberate negative control. This is an MHC that happens to have mutations. It has an R, 2 R's and a Q that mess with the binding, and it sort of repels the receptor. So this MHC won't work, but that's what we expect. This is actually a good negative control. There's no way you're going to get one of these that's going to work with every MHC universally. But working with 3 out of 4, that's probably good enough. You just need to make sure that your patient doesn't have the 4th MHC, because this won't work with the 4th MHC. At least if that's the only game in town. So here's where they actually took this receptor and they challenged it against tumor cells, and they saw tumor killing. The red dots that you see on the far left are tumor killing, and I drew lines. The red arrow points to the red line, which means you add T cells and the tumors don't survive. You see the tumors die off because the T cells are killing them. And this works for the red line and the blue line. You see them both going down. Those are 2 different MHC molecules with the same peptide. They also did a cool experiment where, they did all sorts of experiments where they added the peptide. They had the peptide not there, nothing happened. They mixed in the peptide and it started to kill the tumor, things like that. It's all working, and this does appear to be a pretty good paper. So it looks like it works, and again, they showed that it actually will eliminate tumors in mice, so it will at least work in that mammalian species. Hopefully it will work in ours. So that's an example of a receptor, their CAR receptor, that will cross-react with a peptide across many different MHCs. That's our example 1 of cross-reactivity. And now I'm going to have 4 more examples real quick of what do I mean when I talk about cross-reactivity. Let's move on to the next one, number 2. One TCR binds the same surface presented by different peptides from different diseases. In this case, you can have one disease, teach your TCR how to react, and then that same TCR can react to a totally different disease because the peptides are similar. This is this example, and this is actually what got me started on this thinking a long time ago. This is by Mark Davis's lab. And I actually went to an immunology conference and I saw him speak, and it sort of blew me away because it makes perfect biochemical sense. These peptides are so short that they're only presenting really the residues that point up, right? So they're only presenting like 30 to 50% of the residues that are even touching the TCR. Doesn't it make sense that there would be a case in some case where one disease presents a peptide and educates a TCR that will react against a certain sequence, and that same sequence, just by chance, shows up in another disease, and you get 2 for the price of 1. You get one T cell that was raised against one disease, and it protects you against another. Mark Davis found out there were many cases like this, and it can be traced down to which residues are pointing up and which residues are pointing down. As long as the residues pointing up are similar, you can have the same TCR being activated by two very different diseases if they present similar peptides. Okay, so this is where some of the data starts. Maybe you can tell, I'm not sure even what all these different diseases are, but these are different microbes that cause the disease. A. tropicalis and A. thaliana, entirely different organisms, different proteins, but their peptides look a little bit the same. Look right here, they have FSAL in the middle, and they're different on the other end. But apparently the similarity between these, having this sequence on the N-terminal side of the peptide being similar, means that in 3 different cases, you have T cells that will recognize one, will also recognize the other one. This is what we're talking about cross-reactivity of peptides. And there are several other cases, and sometimes you can trace it down to the exact sequence, sometimes you can't. But the thing is, this exists, and you can trace it down. This is where Davis's study started. He had 5 TCR clones, and these were all, by the way, originally raised against HIV peptides. And then they found out that they would recognize these peptides from weird bacteria. They're like, why is this? And they started to look at the sequences, and they started to realize, oh, the sequences are similar, even though the diseases are very different. Just because peptides are so short, you're going to get that way. So children don't have as many different T cells yet. Maybe the cross-reactive T cells are one of the things that changes you from being a child. You get sick with everything. But then because you have cross-reactivity, maybe your T cells start to recognize even things they've never seen before, if it has a case like this. In this case, somebody who had these T cells against HIV would also have at least some protection against A. tropicalis, which is a tropical disease. Here's a more focused example where you can see the sequence similarity very clearly. You can see E, K, M, N, T, Q in all of these. And in fact, these are the H1N1 bird flu peptides. And so you see that those peptides are similar. It makes sense that they would cause a similar TCR reaction. But the ones on the bottom are very different diseases, but similar peptides. And you can see the similar peptides in that case are also causing, at least in one case, they're reacting with both of the other T cells. And in the other case, one of the T cells reacts and one of them doesn't. One of them cares and one of them doesn't. So the main thing is, these are very different diseases, but similar structures. And the structure is what's causing the cross-reactivity. Because the system is accidentally recognizing something beyond what it should, it's actually in this case giving you extra protection. It's not always good necessarily. Sometimes this cross-reactivity probably causes an autoimmune response. But the main thing is, on the whole, does it protect, does it hurt, or does it help? Well, it changes things, is all I'm sure about. And it means that this can happen. Good things can happen, as well as bad. So example number 3. B-cell epitopes, if they're similar enough, can be cross-reactive too. Here are some cases where you have SARS-CoV-2 compared to the 4 common cold coronaviruses. CCCs is what they call them. And you see the ones underneath, they list all 4 common cold coronaviruses under SARS-CoV-2. And remember that I said that these are 60% similar, and for some short peptides, they're even more similar than that. You can see that these peptides are 80 to 90% similar for the ones on top. And so this is the region, this is a B-cell epitope where the antibody will bind this part of the protein. And if it binds this sequence on top in SARS-CoV-2, if you look at the sequence in the NL63 cold virus, you see that it's very similar. It makes sense that it would be cross-reactive. And it looks like that is actually happening. The antibodies are binding, when that makes sense. And the real question is this even good or bad? Sometimes the antibodies can bind and cross-react, and it could be bad for you. But sometimes it can be good. And it looks like in this case, they found out that if you develop the antibodies, some antibodies against common cold will give you some protection against the progression of COVID, whether it's going to progress to be mild or severe. Here's a case where they put all the viruses together, and I just want to point out, it's kind of hard to see, so I apologize for that. But the red sequences on the top, like the top lobe of the tree, and the blue and the blue-green lobe at the top of the tree, sort of at 12 o'clock and 2 o'clock, those are the common cold coronaviruses. Those are in red on those lobes. SARS-CoV-2 is in the orange lobe, which is sort of at the 8 o'clock position. So it's not directly related, but it's sort of a cousin. It's not a brother or sister, but it's a cousin to the common colds. And because of that, there's enough sequence overlap where some of the same proteins can produce antibodies that will react. And that's what they tested in this study. And remember that we had a fourth example where we talked about how SARS-CoV-1 and SARS-CoV-2, those are 90% similar. So it totally makes sense that if we go to our freezer and we pull out the SARS-CoV-1 antibodies, some of them might bind SARS-CoV-2. In fact, here's one antibody. We can see the sequence where it binds. And yeah, it's not identical, but it's similar enough to be recognized. And so they found that, I mentioned that earlier. This is a specific study about SARS-CoV-1 antibodies that will bind SARS-CoV-2. Here's a sequence, here's another sequence comparison. You can see all the blue shows how similar CoV-1 and CoV-2 are. And over there, the gel, or it's actually a blot, on the left side is going to have, it's an anti-SARS S1 antibody on the bottom there. And yet you see, if you look at the big bands, you see really big bands where it's binding SARS-CoV. But right next to that SARS-CoV-2, you see a faint band at the right spot. So it's not binding as tightly, but it is binding. You can measure it by this blot. The main thing I want you to see is down here in the legend. It talks about how this SARS-1 antibody is polyclonal. That means that it's a bunch of antibodies with a bunch of different sequences, and we just pulled them all out of the animal, and then we purified them. So we know that there's a mixture of them. Probably what's going on here is you have a few of the antibodies in that mixture that are highly reactive, and most of them don't react at all. The other possibility is you have a lot of them that react a little. And I guess that's a possibility. I just think it's less likely. But when you have a monoclonal antibody, that means one sequence, one antibody. You can get down to a crystal structure. You can say it is this sequence right here. This is a little more complicated because it's a bunch of antibodies all together. It's just all the antibodies that you pulled out. So know the difference between monoclonal and polyclonal. For the purposes of this study, the polyclonal antibody shows binding, and we just know that there's a bunch of different sequences. We don't know which ones are the sequences that are binding without further study. And finally, number 5, this is the one that's really cool to me. You know how I'm always talking about getting your vaccine because you don't know what good it can do. If you put all these together, you can say, well, if I can get one disease and have cross-reactive T cells, maybe I can get one vaccine and have cross-reactive T cells against other diseases. And it looks like that actually can happen. Again, how much? Is it good or is it bad? We're not sure yet, but it looks like in this case there's at least a weakly good response, where if you get the MMR vaccine and or the Tdap vaccine, those antigens have some structural similarities with COVID-19 causing disease, with the SARS-CoV-2. And so that means that if you're immunized for MMR, you could have some T cells that will actually react and some immunity that will actually protect. And they actually, this isn't just at the level of cells. They actually took this out to see if the vaccinations correlated with severity. And they found out, at least in this study, that they did. People who had their MMR Tdap shots were actually protected against, they had less severe COVID when they got COVID. Now, they didn't show the structures in here, so what I would want you to do if you were reporting on this, I want to see the structures. And so they have right here this paragraph where they say these 5 different things tell us the structures that show us the sequence similarities that could be giving us this cross-reactivity. That's what I want to know more about, but we've already gone on too long. I just want to say I'm convinced that this is real. I don't know how strong it is, and you can always say, oh, that's just how the cells work, but how do they work when you're in a whole human? And that's very valid. But at least we know that there could be something good going on. And again, it could be bad. You can have autoimmune reactions. There's something called original antigenic sin, which is actually a term, it doesn't really make theological sense, and I wish they would retire it, but that's what people call it in the literature. So you might come across something like that. So, I'm going to have one more lecture on this, but it's going to tell a story about cross-reactivity and about COVID and about COVID worldwide. And so it's an interesting little story, but it involves science, and I'm going to look up some of the data for that for you. For presentation number 4, I want you to do a presentation for me about cross-reactivity. Or, of course, you can follow the original instructions, which were about evolution and predictability. If you want to do cross-reactivity, give me an example like these 5. Notice that I'm always showing you structure at some point, structures and or sequences. I want you to show me some biochemistry. So don't just give me something vague where you don't show me the biochemical rationale for why would this be cross-reactive. I want to see similar sequences. I want to see shapes, antibody recognition sites that have structural similarities. But I'm really interested in this more than anything else. So if you're interested in it too, follow that interest, find an interesting story, and tell it to the class. You have 10 minutes to do that. Show us some structures. So yeah, I'm looking forward to see which of these options you choose. Please do sign up on with your particular title of your paper again, so we can make sure we don't have any overlaps from people. And thank you for listening. This is the end of the structural immunology. This will be on quiz 2, and I will see you all for your next presentations on Wednesday.